Hello friends, today in this video we are going to discuss about the skill test or trade test or practical test or job oriented practical test on biological science or life science for government jobs. Okay, so in many government jobs for biological science or life science, they said that they mentioned that skill test or practical test or trade test you have to perform after written test. Okay, so for that skill test or practical test, what types of questions you have to, what type of practicals you have to perform. So all these things we will discuss in this video one by one. Okay, so at first we have to know what are the subjects which are present in biological science or life science. So these are the subjects are zoology, botany, microbiology, biotechnology, biochemistry, food technology, agriculture, molecular biology, genetics, all are involved in or present in biological science or life science. Okay, so if you are graduate in any of these subject, you are eligible to give exams in government exams in under the biological science. Okay, so move on to the next, that is, at first before going to the skill test or practical test, at first we have to know in which type of post we can get in government jobs for biological science. So at first we have to discuss about this. So that is laboratory jobs in biological fields. So which are the laboratory jobs are present in biological field in government sectors. Okay, so we have distributed it in two ways. So first one is lower grade jobs and the next one is higher grade jobs. So under these lower grade jobs, which are, that is the level 2, level 3, level 4 jobs, so this type of jobs are there, that is laboratory assistant or laboratory attendant post, next laboratory technician post or testing personnel. So all of these are lower grade biological field jobs in government sectors. Okay, and for specific jobs or higher grade jobs, that are the food safety officer job, scientific analyst job, technical assistant job, or scientific assistant jobs. So many types of specific jobs are there for biological fields. Also, that is a microbiologist post or biochemist post or food technologist post, all are there in biological fields. Okay, so many specific and not specific, many posts are there for biological fields. So now in government sector, the government exams are with the written test and skill test or practical test. So for written test, they mentioned the syllabus, but for practical test or skill test there is no syllabus. So we have they can give anything to perform in practical. So what are the probable questions or probable practicals you have to perform in this skill test or practical test? Here we will discuss. So first we will see the topics of skill test, trade test or practical test or job oriented practical test. So two topics here we discuss, that is identification of lab items and instrument. That means many types of lab items or instruments which are used in labs or biological labs specifically, that we have to identify. They will show you and you have to identify this, and we have to say what are the use of those instrument or items, lab items. Okay, so that is the one thing. And next is the practical to be performed. So what are the practicals? Say they will give you some practicals and we have to perform those practicals to qualify the skill test or trade test or practical test. So at first we will discuss the identification of lab items, and then we will show some practicals which we have to perform here. Okay, so the first thing is the identification of glassware. So they can give you to identify the specific glasswares and their use in those biological labs. So first thing is the test tube. So what are the role of test tube and where we can use this we have to mention. And at first they will also show this picture, or also you can they will show the test tube, and they will say to mention what is this glassware called and what are the use of this. Okay, so next is the measuring cylinder. So what is the use of measuring cylinder? We have to say that is the to measure the particular volume of solution or water. Okay, next is the conical flask. So where it is we can use, so conical flask is used for mixing different type of solution or reagents or medias here, and or for autoclave you can use conical flask. Next is beaker. So here also you can collect the volume of water or volume of reagents or solutions or perform any reactions. Okay, next is glass pipette. So by glass pipette you can transfer minute amount of samples from one solution container, one container to another container. Next is volumetric flask. Another many type of flasks are there, that is Florence flask, boiling flask, separating flask, many other flasks are there. And next is slide. So slide is used for staining purpose and to see the microorganisms under microscope. And next is glass funnel. Okay, so we have to mention all the use of this glasswares in lab purpose. Okay, so the next question or next practical we have to perform, that is identification of lab items and their use. So lab items which are present in laboratory, that is first thing, that is filter papers. So what is the use of filter papers? To separate the impurities from a chemical solution. Okay, so separate the impurities or separate one thing from another from a solution. So in this purpose we can use filter paper, and we have to also mention or remember the pore size of different types of filter papers and their make. Okay, so next thing is the 70% ethanol. So 70% ethanol is used as a disinfectant to disinfect the workplace. So where you are going to perform your test or experiment, so you have at first you have to disinfect those place, and then we have to work in biological labs. So for this disinfection we have to use 70% ethanol. So next is spatula. So spatula is used to transfer the powder form or any form of material or reagents to measure under weighing balance. Okay, so next thing is the syringe filter. Syringe filter is used to sterilize the microbiological samples or microbiological solutions which are which we cannot autoclave. Okay, so this type of solutions or serum or anything we can sterilize by this syringe filter. So their filter pore size is 0.45 micrometer or 0.22 micrometer, and cellulose nitrate membrane is used here, or different type of membrane filters are used. So all information we have to know about this. Next thing is the parafilm. So parafilm is used to cover or wrap any petri plate or any microorganisms plate to remove the contamination or remove the air contact or moisture contact. Okay, so next thing is cryovial. So cryovial or cryotubes are used for cryopreservation, or if you are going to preserve microorganisms in a -80°C, so you have to put them in a cryovial and then you can preserve them. Okay, so next is the Bunsen burner. So Bunsen burner is used to incinerate the microorganisms which are already inoculated by inoculation loop. Okay, so for incineration purpose we use Bunsen burner. Okay, so next thing is the inoculation loop. So inoculation loop is used to inoculate the microorganisms from one plate or from one solution or from one media culture to another. Okay, Next is the Petri plate. So Petri plate is used to grow the microorganisms here. And another is Eppendorf tube. So here it is used in centrifugation or any different type of molecular biology uh, experiments. Okay, so move on to the next. There is next thing which they can ask you in skill test or practical test, that is identification of lab equipments and instrument and their roles. So what are the lab instruments or equipments are available in a biological lab? You have to identify them and we have to say their roles in those lab. So first thing is the autoclave. So autoclave is used for sterilization, that is the moist heat sterilization, where water and heat is used, okay, for sterilization. That is a complete sterilization. So you have to know the principle of this autoclave. That is it is run in 121 degrees Celsius for 15 minutes for 15 lbps pressure. Okay, so and the precautions what you have to take before and after starting the autoclave, so all we have to know. So next thing is the colony counter. So colony counter used to measure the colonies or count the colonies. So here we have to put our Petri plate. So we have to put our Petri plate and the colonies are counted by a pen. Okay, and all the count you can see here by digitally. And we have to, here a magnifying glass is there, so to see those colonies elaborately. Okay, so next thing is the weighing balance. So weighing balance is used to weighing particular reagents or medias. Okay, so next thing is a pH meter. So pH meter is used to measure the particular pH of a solution or reagent or any microbiological media. Next thing is the fumigator. So fumigator is used to fumigate a room. Suppose a room where any type of organisms or microorganisms are work in a laboratory. So you have to fumigate the room at least once in a week, so that the room is not contaminated with many types of microorganisms. If the microorganisms, if the room is contaminated with many types of microorganisms, then it can hamper your experiment or test. So that's why we have to perform fumigation. So at first here we have to add the fumigating agents, and here a fan is present, so it circulate all the air or fumigating agents throughout the room, so that the room can be sterilized, okay, or free from any microorganisms. Okay, so move on to the next one is the laminar airflow or biosafety cabinets. So all these laminar airflow or biosafety cabinets are used to get a sterile environment, so you can perform a experiment under this sterile condition. Okay, so next thing is a vortex shaker or vortex mixture is used for mixing any solution. Okay, next thing is a centrifuge machine. So centrifuge machine is used for centrifugations, and here the centrifuge tubes are present where you put your solution, and after centrifugation, sorry, for centrifugation we have to set the rpm and also the time. So after centrifugation, you if your requirement is the pellet, then you separate the supernatant, and if your requirement is the supernatant, so you take it and uh, separate the pellet. Okay, so this is the centrifugation process. Next is the anaerobic gas jar. So anaerobic gas jar is used in a lab for anaerobic, creating anaerobic condition for growth, for growing the microorganisms which are anaerobic microorganisms. So for growing anaerobic microorganisms, we use anaerobic gas jar. Okay, where a gas pack is present. So here we have to put a gas pack which produce or which eliminate the oxygen from here and produce a anaerobic condition in this chamber. Okay, so next thing is the minus 80 degrees Celsius deep freezer, which is also known as ultra low temperature deep freezer. So it is used to preserve microorganisms at minus 80 degrees Celsius for long time preservation. Suppose for 2 years or 3 years we can preserve those microorganisms in this minus 80 degrees Celsius by producing the glycerol stock in a cryovial. Okay, so move on to the next equipments, that is the BOD incubator or normal incubator. So incubator is used to give a particular continuous temperature. Suppose you require 37 degrees Celsius for growing a microorganism. So we have to put those culture in this incubator and set the temperature 37 degrees Celsius. So for 37 degrees Celsius for 1 day or 2 day, it will give a continuous 37 degrees Celsius temperature, okay, for growing microorganisms. Next is hot air oven. So hot air oven is used for sterilization, heat sterilization. So suppose for glasswares which are used in laboratory, these glasswares can be sterilized here by hot air oven at 160 degrees Celsius for 1 hour. Okay, so by this technique we can sterilize the glasswares in lab. Next thing is the hot plate. So hot plate is used for solutions we have to heat to boil. So for that reason we use hot plate. Next is the water distillation unit to produce distilled water, which are used in any type of biological lab. Next thing is membrane filtration unit, where a membrane filtration is present and the solution is passed through this membrane filter, and all the microorganisms which are present in this solution, it will trapped in the membrane filter, and then we have to collect the membrane filter and place it into a petri plate or agar plate, specified agar plate, and then we can see the growth on the petri plate if the microorganisms are present in that solution. Okay, so this is the membrane filtration technique. Another equipment which is used, that is water bath. So water bath is used to keep the solution or any media at under 100 degrees Celsius for 15 minutes or half hour or 1 hour. Okay, so next one is the shaker incubator. Shaker incubator is used when a microorganism require their for their growth, they require a particular temperature with continuous shaking. So for that we use shaker incubator. Next thing is a spectrophotometer. So for spectrophotometer we can see the value of OD of any solution. Okay, so for microorganisms we can collect the their growth. We can evaluate their growth by their OD value through this spectrophotometer. Okay, so next thing we will see, that is bio-waste dustbin color code and use. So in every biological lab different bio-waste are generated, and for different bio-waste different color are given for different waste. Okay, so for yellow bags or yellow dustbins are used for infectious waste or bandage, gauze, cotton, other objects which are contact with the body fluids or human body parts, placenta, etc. are collected in the yellow bags or yellow dustbin. Next, in red bags or red dustbin, we will collect the discard the plastic waste such as catheters, injections, syringe, etc. In blue bags, all types of glass bottles are collected or broken glasses are collected, and in black carry bags or black dustbin, we collected all syringe or metal particles, any sharp and metal particles, blades, syringe, anything. Okay, so next one is the practical to be performed. What are the practical we have to perform? So first practical, that is weighing balance or weighing in a weighing balance or analytical balance. So suppose you have given a condition, that is prepare a media solution of 100 ml containing beef extract, peptone, NaCl, and distilled water is 1000 ml. So we have given the instruction that this all this quantity are for 1000 ml, but you have to prepare 100 ml solution, so you have to convert it for 100 ml. So we have to add beef extract 0.3 gram, peptone 1 gram, NaCl 0.05 gram, and distilled water 100 ml. Okay, so you have to measure all in this weighing balance and mix it in a conical flasks. Okay, so in this way we have to perform this practical or skill test. So move on to the next practical which you have to perform in a test, that is how to operate autoclave and what are the precautions you have to need it, and time, temperature and pressure we have to set there. So for operating autoclave, at first you have, what are the precautions you have to need it? So at first you have to check the water level should be up to the electric coil or boiling, that is the heating coil. So water level should be upper side of this coil. Okay, next you have to wrap all the materials which you have put it in this autoclave, that means microbiological media or reagents or solutions you have to put it here. And if there is a bottle is there with a screw cap, so you have to loose the cap here and put it into the autoclave. Okay, so many types of precautions you have to need it before that, and after that we have to set the temperature, time and pressure in those autoclave. Okay, so all these things you have to perform by your own, and you have been given the marks on the basis of your knowledge of practical, okay, or handling machines. So move on to the next practical, that is buffer preparation. So you have to given a chart and you have to told uh, to perform a buffer, prepare a buffer, that is you have given all the instruction, that is you have to add this amount of base, this amount of acid, this amount of water, and we have to add all these things and prepare a buffer. Okay, so this is a practical. Next thing is a micropipette handling. So we have to set we have to you have instructed that we have to set volume for 600 microliter and take the solution from container A to container B and eject the tip to the discard. So what you have to perform? So at first we have to take 600 microliters. So for taking 600 microliter solution, at first we have to take the micropipette, that is 1 ml micropipette or 1000 ml micropipette. Okay, so for 1000 ml micropipette, here the digit is showing, so you have to set the volume 600 microliter. So for that you have to set it as 060. Why? Because if it is a 1000 microliter pipette, then it is showing 1000. Okay, and it will only show 3 digit. So upper 3 digit, this 3 digit you can see in this micropipette, but in the lower thing you can show, you can see. Okay, so for if you have said to prepare 1000 microliter, then we have to set 100. And if you are said to set 900 microliter, we have to set 090. And here you have instructed to set 600 microliter, so you have to set 060. Okay, and after that you have to transfer 1 microliter, sorry, transfer 600 microliter from container A to container B, and then we have to eject through this ejector. Okay, so this type of practical we have to perform. So next thing is pH measurement. So you have to calibrate the pH with pH 4 and pH 7 and measure the pH of the solution provided. So we have given a pH 4 solution, pH 7 solution and a unknown solution. Okay, so at first we have to calibrate the machine with your pH 4 and pH 7 known solution, and after that we have to perform or measure the unknown solution in this pH meter. Okay, so move on to the next practical, that is microscope handling. So how you handle the microscope. So you have instructed to set the slide at 100x objective lens and take a picture of a well defined field. So at first we have to take the slide and cover those smear with a cover slip, and after that we have to add the oil or for oil immersion, because here 100x is instructed. So after that we have to set or focus the knob in the microscope and take a well defined picture for a suitable field. And also you have to remember all the points, that are the coarse adjustment knob, fine adjustment knob, or diaphragm, which is diaphragm, which is condenser, which is objective lens, which is ocular lens. So all these things we have to know for the microscope. So all depending on all this knowledge you will be given the marks. Okay, so the next thing is streaking on agar plates. So we have instructed that perform a streaking within laminar airflow with proper precautions for isolation of pure culture. So you have given a solution or a petri plate where microorganisms are present or cultures are present. So where you are given a agar plate, so where we have to perform the streaking, that is a four-way streaking. This type of streaking we have to perform. Okay, so the next thing is the DNA loading and gel electrophoresis. So we have instructed to add 10 microliter of DNA to the well and prepare electrophoresis assembly to start. So at first here the agarose gel and here the well is present. So in the well we have to add the 10 microliter of DNA by a micropipette. Okay, and then we have to prepare the electrophoresis assembly. So where the positive and where the negative end is should be present, what are the where the cathodes and anodes should be present. So all these things we have to assemble here. Okay, so next thing is the next practical is gram staining. So here we have to heat fix the organism on slide and perform the gram staining. So at first we have given a solution or culture and a slide. So you have to put make a smear here, and then you have to heat fix it, and then you have to perform the gram staining. That is, at first you have to add crystal violet, then iodine, then alcohol uh, wash, then safranin. So in this way you have to perform the gram staining procedure. Okay, so the last practical which can be asked in a skill test or practical test in a biological lab, that is, prepare yourself to work on laminar airflow and what are the precautions and safety measures we have to take. So before entering in a laminar airflow for work, what precautions you have to take, that means you have to take apron, and next you have to wear the gloves, surgical masks, goggles, everything we have to wear, and then we have to clean your workplace by alcohol or 70% ethanol. Okay, so after then we have to perform your experiment. So here a experiment they have suggested, that is inoculation inoculate one loopful of organism into agar slant. So here agar slant is present, and here one loopful of culture you have to collect and add it or streak it into the agar slant. So this type of practicals you have to perform in a skill test or practical test or trade test for biological jobs, okay, in a government sector. So these are all the practicals you have to perform, and depending on your skill or depending on your knowledge on practical, you will be given the marks. Okay, so thank you for watching this video.